To the entire audiobook listener who likes to listen audiobook, please subscribe my channel The Learning Team in order to get more useful information. Chapter 2, Metabolism, Energy in the Cell. How a cell transforms matter and energy to do work is called metabolism. The goal of this lecture is for you to understand how cells turn sunlight or food into a form of energy that can be used to do the necessary work. Number 1. Photosynthesis, our solar-powered world. Photosynthesis, or the conversion of sunlight into sugar and other chemical energy stores, provides sustenance for almost every living thing on Earth, either directly or indirectly. Even the fuels that we burn to heat our homes and power our cars have their origins in plant and animal life that put the sun's energy into storage hundreds of millions of years ago. Human cells have evolved without the capacity for photosynthesis. What makes plant cells special? The answer lies in part in their structure. Our ability to move around would seriously impede the ability to capture energy from the sun the way that plants do. Our cells are squishy and mobile, plant cells are much more stable and structured. This architecture allows them to take advantage of certain properties of a specific molecule called chlorophyll. To turn sunlight into energy, plant cells take carbon dioxide molecules from the air and water molecules from the soil and add the energy of sunlight to make glucose, or sugar. Then, they either use the molecules of extra oxygen and water in other reactions or expel them for our consumption. This reaction is essentially what our cells do, but in reverse. We add oxygen and water to glucose and create the energy that our cells need to work, plus the byproducts, carbon dioxide and water. Photosynthesis largely consists of two components. First, the light reactions capture energy and require sunlight. Second, that energy is then used to build glucose and other molecules in a process known as the Calvin cycle. These are also called the dark reactions, because they don't require light. Both of these processes happen in an organelle called a chloroplast, which is unique to plant cells. Chloroplasts are similar to the mitochondria that power animal cells. All eukaryotes have mitochondria, but plant cells also have chloroplasts. The structure of chloroplasts is the key to their success. Surrounding the organelle is an envelope of two membranes, like the nucleus in a eukaryotic cell, and inside it is a fluid called the stroma. Within the stroma is a third membrane system, which is made up of sacs called thylakoids. Thylakoids are often stacked in columns called grana. This structure, while highly efficient, simply would not be possible if plants were mobile the way animals are. It is flexible enough to bend a bit, but it is just too rigid for walking, running, and jumping. Chlorophyll, which is pigmented green and is the star of the photosynthesis show, is found in the thylakoid membranes. The light reactions convert solar energy to chemical energy, and this happens in the thylakoids, where the chlorophyll is. Photosynthesis is the process by which light is turned into food. First, the light reactions capture energy from the sun in the form of photons and then use it to make adenosine triphosphate, ATP, and nicotinamide adenine dinucleotide phosphate, NADPH. Then, the Calvin cycle, or the dark reactions, uses these products to create sugar with the help of carbon dioxide. The sugars then supply the plant with chemical energy and carbon skeletons so that the plant cells can manufacture all the molecules they need to perform the functions of life. The mitochondria in plant cells use the products of photosynthesis as fuel, and the excess sugars are stored in various parts of the plant, like in seeds, roots, and fruits. Humans and other animals are attracted to plants. They eat their seeds and fruits and disseminate the plant's genetic material so that new plants can grow, because, of course, plants can't move around by themselves. Perhaps most importantly, one of the byproducts of photosynthesis is oxygen, which animals like humans need to survive. Plants use up carbon dioxide, which is emitted by animals during their own metabolic processes. Without photosynthesis, life could not survive on Earth. In addition, metabolism in animals helps power photosynthesis by providing carbon dioxide. Number 2. Cellular respiration. Cells have multiple jobs, and of course, different types of cells have different subspecialties, from muscle control to information signaling to detoxification and development. All of these jobs, and the factories that create the products needed to accomplish them, require fuel. In animals, food needs to be ingested and converted to energy so that the protein factories can be powered. This process of converting food into fuel is called cellular respiration, which is photosynthesis in reverse, taking sugars, adding oxygen to them, breaking them down to release energy, and expelling carbon dioxide as a byproduct. Just like in the internal combustion engine in your car, your cells mix hydrocarbons with oxygen and let out an exhaust of carbon dioxide and water. Moving electrons around releases energy in organic molecules, energy that is ultimately used to build ATP, so that the energy can be stored for later use. Cellular respiration can be divided into 3 main phases: glycolysis, the citric acid cycle, and electron transport. Glycolysis is the process by which sugar, in the form of glucose, is split into 2 molecules of a compound called pyruvate. It's a 10-step process, and the end products are pyruvate and 2 molecules of energy storing ATP. After glycolysis, pyruvate migrates to the mitochondria and enters the organelle. There, oxygen is added to a reaction that produces a compound called acetyl coenzyme A, acetyl-CoA, that then enters the citric acid cycle. Like the Calvin cycle in photosynthesis, the citric acid cycle, or Krebs cycle, can be thought of as an assembly line that manufactures specific products. Acetyl-CoA enters, and then there are 8 steps that eventually result in the release of ATP and a return to the original state so that another acetyl-CoA molecule can pass through. After the Krebs cycle, the last step in cellular respiration is what is called the electron transport chain, in which electrons are passed along in a series of reactions, gradually releasing energy that can be used to make more ATP. The building blocks of the chain are located in an internal membrane within the mitochondria so that the different molecules in the chain can be held closely together. In this way, the structure of the mitochondria is reminiscent of the chloroplasts. Ultimately, the electrons are passed down to oxygen, the final electron receptor. Because of its polar properties, oxygen draws the electrons toward it along the chain, and because we breathe in and out all the time, there's plenty of oxygen to go around among all of our cells. But the electron transport chain is just the first step in this last phase of metabolism in the mitochondria. In fact, the next step is likely the most important. whereas glycolysis and the citric acid cycle produce about 2 net molecules of ATP for use elsewhere during each cycle. Oxidative phosphorylation, or the combination of the electron transport chain and a process called chemiosmosis, produces 26 to 28 molecules of ATP. In the chemiosmosis process, energy from the electrons in the transport chain is used to pump protons, or hydrogen ions, across the membrane, and then they move back through an enzyme called ATP synthase. The flow of protons back across the membrane provides the energy needed to form ATP. The remaining electrons and protons at the end of the pumps are joined with oxygen to form water. Number 3. Metabolic engineering. Knowledge of the metabolic pathways, or chemical reactions in cells, has been used extensively to improve our lives. Harnessing a cell's metabolism to build products is called metabolic engineering, and the ultimate goal is to develop cost-effective and ethical ways to produce valuable substances in large amounts. Current metabolic engineering techniques involve using well-known, safe microorganisms on an industrial scale to produce bulk chemicals such as those used in fragrances, essential oils, vitamins, agriculture, and many more, but the future entails tailor-made designer cells that might make synthetic organic chemistry production obsolete. One of the first major success stories involving metabolic engineering is the production of an anti-cancer therapeutic agent, currently used in the treatment of several types of cancer. In this story, the needs of cancer patients were pitted against the environmental costs of harvesting a naturally made compound. A chemical called Taxol was discovered in the 1960s. It was collected from the bark of a Pacific yew tree known as Taxus brevifolia in a forest in northern Washington state. In the late 1980s, the compound was shown to have a beneficial effect on patients with melanoma, or skin cancer, and ovarian cancer, but in order to treat all the current cases in the United States, the pharmaceutical company would have to destroy 360,000 trees every year. By the 1990s, as the environmental movement was rapidly growing in the United States, there was pressure to create the drug synthetically, saving the trees in the process. The best method discovered took advantage of metabolic engineering and, in the process, eliminated the environmental costs of killing trees. It also uses fewer toxic chemicals in its production than purely synthetic methods, and it uses less energy. Today, the drug Taxol is FDA approved for the treatment of breast, ovarian, and lung cancer, and it is even available in a generic form. All production of Taxol by the Bristol-Myers Squibb Pharmaceutical Company uses plant cell fermentation technology, and there is no longer any need to take bark from trees. Metabolic engineering is not widely used in the pharmaceutical industry, but it has also improved production of some of our favorite vices, cheese, beer, and wine. Thank you for listening.